Our next speaker in the session is Joon-Hyun Song. Join me, welcome.
Good afternoon. I'm Joon-Hyun Song from Stony Brook University. I'm working with Dr. Med Damake. And I would like to talk about, first, I want to thank the organizer for giving me a great opportunity to present my preliminary works. My work today, I wanted to present about the microenvironmental variability, endormancy, and the maintenance of intratumor heterogeneity. So let's start with the cancer ecology. Cancer cells do not evolve in a vacuum, but in the interactions with the environment and other cells, as you can see, that creates a great heterogeneity. And what I want to mention that we are working on spatial temporal spheroid model under radiation that forms this beautiful heterogeneity across the spatial and also temporal. And I am interested in more on temporal variation in our human body. We are not living in a static world. Our body are very dynamic. Cell experience regulated and controlled and predictable rhythms like circadian clocks or hormonal changes, but also experience uncontrolled, unpredictable variations driven by exogenous uncertainties. For example, this two examples of cycling hypoxia measured in tumor, that we know this unpredictable changes happen in the tumor. So, like I said, the tumor heterogeneity is, we all know tumor is heterogeneous, and the source of tumor heterogeneous came from genetic mutations, epigenetic alterations, and nosy gene expressions, also from spatial distributions. But I want to focus on temporal variation because that's very tricky to study by only by experiments, because in many cases biological experiments are destructive, which means you are gonna lose the exact sample you wanna study over time, but also tracing those temporal fluctuation in tumor microenvironment in vivo is very challenging. That's why mathematical and computational study on this temporal variation is important. And one thing I'm interesting how this temporal variation can create heterogeneity is through a mechanism called storage effect. The storage effect is a mechanism to explain coexistence of multiple similar species within the same ecological community. To have this kind of storage effect, there needs three essential factors, which one is species-specific environmental response, which means species will react or respond different in different environment. And also there are covariance between environment and competition. And the third, which I'm gonna more be focused on this study, is how the buffered population grows. And one of the way of buffering is dormancy. I would like to briefly explain how storage effect occurs. In this, for example, imagine there's a two type of plants, red and green, red and blue. In good condition, both can live. You can see red are growing slightly better, but they both red and blue plant can have a seed bank, as you can see under the ground. And when the bad condition came, most of them die. One slightly resilient, one blue one might survive. But after this harsh environment. When the good condition came back, because they're seed banks, the red and blue plant both can grow and maintain coexistence. So why I look into this kind of dormancy is because we also know that cancer cells, some of the cancer cells can go into what they call quiescent state. They don't divide, but they also don't die that much. So this is one way the population can have buffered population growth and can display a storage effect. So my big question is how temporal variation in microenvironment affect human heterogeneity. And the more specific focus of this study was, is there a difference in maintaining heterogeneity with and without dormancy, and how the outcome change with fluctuation patterns? And is dormancy advantageous under unpredictable random fluctuations? I use ABM. I don't think I need to explain what this ABM is. But I would like to explain very, very briefly the base model framework I worked on. This is my base model framework is very simple. Each cell has a growth parameter per environment. They have two values per environment, which has probability of division, probability of death. And also one additional thing is a cell state. Cells can have multiple states, not simultaneously, they can change, but different states have different growth parameters, and they change stochastically. I put those state transition to model dormancy, and each time step, as you all know, updated states first, and then updates is fate, which is division, death, or state. I start with a simple parsimonious model and increase complexity one by one to see the effect of dormancy. First, I looked into two lineages model. Lineage 0 and 1 favors environment 0 or 1 respectively. Their growth rate in good or bad condition is same, meaning the lineage 1 growth rate in environment 0 will be same with the lineage 1 growth rate in the environment 1. As you can see in constant environment, obviously, quickly lose its less adapted lineages. Just one lineage survive. There's no coexistence, no heterogeneity. However, if there is a periodic fluctuation or random fluctuation, two lineages can coexist. Although in random fluctuation, it's much unstable. And as you can see, without dormancy, if unfavorable environment persists, one of the lineage might disappear. However, with the dormancy can buffer cells not to die off in unfavorable conditions. With left one with probability to go dormant, 1 per step, which means 1 cell out of 100 cells will go dormancy. That small value can save population to exist longer than without dormancy state. And if you increase those probability much higher, you can see a very different pattern. So after looking with these two lineages, I looked up to with multiple lineages. This is how I make a population. I had to model, like I cannot just randomly sample those two growth parameters, because if I just pick randomly, we can get species lineages that's optimal to both conditions, which will lead to just that one lineage will survive. So I model simple linear trade-off and picked from those Pareto frontiers. And dormancy was implemented same in two-lineage model. There are state transition in unfavorable conditions. It goes dormant state, which has very low division and death probability, and just stays. So it's obvious in constant environment, the population will become in like a biased state. But in fluctuating environment, those growth parameters can have more heterogeneous. On the top is like even in periodic fluctuation, and you can see in random fluctuation, more lineages can survive. This is without dormancy. If you add dormancy, even in constant environment, because some cells go dormancy, you can see after 1,000 steps, there are more lineages, but if we keep growing this constant environment, they will eventually lost. But in fluctuation environment, both in periodic and fluctuating one, they keep a much higher heterogeneity after these elections. I looked up into Shannon Diversity Index over time. After this green and red line are showing the dormancy model with the dormancy. You can see they are after 1,000 time points, they are still having slightly higher heterogeneity than the population without dormancy. So as a conclusion, periodic or random fluctuation is required for coexistence of two or more lineages, and temporal environmental variation helps population to maintain heterogeneity, and also dormancy supports population to maintain coexistence and heterogeneity better in random fluctuation. Of course, I have to do much more work on this model. I have to adjust the parameters in multiple lineage models. I can basically pick up more biased population or some more interesting population. And also based on Chiasson's coexistence theory, I can exactly measure how the strengths of storage effect and correlate with the heterogeneity. I would like to thank my advisor, Mehdi Damaki, and David Basanta, who helped me developing this idea on like storage effect and things. Also acknowledge my lab. And yeah, this is my presentation.
Other questions?
Thank you for your talk. I'm also interested in dormancy, so I had a question about that. But so I think what's interesting about dormant cells is not just the fact that they stay dormant, but eventually they sort of awaken. And in cancer, at least, they turn into a recurrent and often metastasized disease. In your sort of like ecological framework, do you have any idea like what would explain these dormant cells ultimately like waking up and start proliferating again?
For now, my model is just simply by environmental cue. I only model like two discrete environment, like good or bad. So in bad, they can go into dormancy. In good situation, I can change the parameter, but for now, all of the dormant cells wake up at that time point. Like if they enters good environment in my model, they will wake up. But that's definitely interesting things that I can change those parameters to change when they can wake up or not. So yeah, that's very interesting. Yeah.
Thank you.
It's time to speak it again.